12.3 deals with DNA, RNA, and protein synthesis. So RNA is a molecule very similar to DNA. It is a nucleic acid still. The differences are the 5-carbon sugar that is on the RNA is ribose. It has one extra oxygen, and its RNA is generally single-stranded. And RNA will also not be using the ATGC scheme we're used to. It will have a AUGC scheme. So uracil will replace thymine. The ability to copy a single DNA sequence into an RNA makes it possible for a single gene to produce hundreds or even thousands of RNA molecules. And there are 3 main types of RNA: messenger RNA, ribosomal RNA, and transfer RNA. Messenger RNA, also known as mRNA, are those molecules that carry the instructions for assembling of amino acids. Ribosomal RNA, rRNA, makes up a portion of the ribosome, which is the site of protein synthesis, as we know, and tRNA transfers the amino acids to the ribosome. Take a look right here. We have a picture of mRNA starting on the left, in the middle rRNA, ribosomal, and all the way to the right is tRNA, which is carrying the amino acid. Transcription is the process of copying RNA molecules where part of the nucleotide is copied. So during transcription, RNA polymerase will bind to the DNA. So we take our DNA molecule, our regular old DNA in the nucleus, bind RNA polymerase to it, and then bases the mRNA off of what is on the DNA. So whatever the sequence is in the DNA, it's going to use complementary base pairing and form an mRNA based off it. Now, promoters are sections of DNA which specific base sequences where the RNA polymerase binds. So promoters are signals in the DNA that indicate to the enzyme where to bind to make RNA. And similar signals in the DNA cause transcription to stop when a new RNA molecule is completed. Let's take a look right here. We can see the DNA double helix unwinding, the RNA polymerase forming mRNA as it moves along. Now, introns are pieces that are inside the mRNA sequence that need to be removed, and exons are the express sequences that are spliced back together. So those are the good parts. Proteins are made by joining amino acids into long chains called polypeptides. Each polypeptide contains a combination of any or all of the 20 amino acids. The properties of proteins are determined by the order in which different amino acids are joined together. So let's first take a look at this. This is the DNA in blue. The pre-mRNA contains the introns, which are still in blue, and the mRNA is once you've removed those introns. Now, mRNA instructions are called the genetic code. Each genetic code is 3 bases long, and a codon consists of 3 consecutive nucleotides that specify a single amino acid that is to be added to the polypeptide. The codon AUG can either be methionine or it can be the start codon. UAA, UAG, and UGA all represent stop. So we take a look right here. Here's 2 3-base sequences, AUG, the start codon, and AAC, another codon. This is a way to figure out which amino acid the codon codes for. Do not worry, you will not have to memorize this. If you are ever asked which amino acid is being coded for, you will be given something like this. And it's very easy to use. If you have AUG as your codon, that middle circle with the big A, the big U, and the big G, and the big C, basically what you do is you find the A, and you go into that quarter of the circle that's the A, and then you go into the section for U, and you find the G. So A, U, G, and it's methionine. You work your way through. The sequence of nucleotide bases in an mRNA molecule serves as the instructions for the order in which the amino acids should be joined together to produce polypeptides. The ribosome factory is where the sequence is read and put into use. Translation is the decoding of an mRNA message into a polypeptide chain. And takes place on the ribosomes, and during translation, the cell uses information from mRNA to produce proteins. So basically, before translation can occur, mRNA must be transcribed from the DNA and released into the cytoplasm. Translation begins when mRNA attaches itself to a ribosome. Translation occurs at the AUG start. tRNA has a complementary anticodon that matches with the mRNA codon. The ribosome forms a peptide bond between the first and second amino acids. At the same time, the bond between the start codon and its tRNA are broken, and this allows the ribosome to bind to another tRNA. The ribosome moves along the mRNA, binding new tRNA molecules and amino acids. This process will continue until it releases one of the stop codons and the newly formed polypeptide is released and translation is completed. Proteins are extremely important because they make up enzymes which catalyze reactions. Proteins are keys to almost everything living cells do. So if we take a look right here, starting in the top left, that is an mRNA leaving the nucleus after it's been transcribed from DNA. Then we see the mRNA attaching to the ribosome. That's that blue and gold structure. And then those little T-shaped, the little cross-shaped things with the red dots on them are tRNAs carrying amino acids into the particular order. And then those amino acids are going to be linked together to form a protein chain. If we take a look right here, we see the DNA at the top, the mRNA next, and at the bottom we see the amino acid sequence.